I'm Choi from Binghamton. So today I'm going to talk about a multi-anode paper-based microbial fuel cell for potentially powering disposable biosensors. So let me first talk about some background. So as we know, while the top killer disease in developed countries are heart disease, diabetes, complications, and cancer, the developing countries are suffering from the infectious disease. So while we don't need to worry about the infectious disease for our newborn babies, it is completely different story in the developing countries. So effective diagnosis of infectious disease is the first step for the appropriate treatment. So people are developing a low-cost and disposable biosensors. And what is the best material for those disposable biosensors? I believe that is paper, because paper is low-cost and combustible, so it is disposable. And it has the ability to weak a fluid, so we don't need any external pump. So it has high surface area and porous and biocompatible and flexible. So Dr. Whiteside group and many other groups are developing those paper-based biosensors, but the problem is the primary detection technique for those devices is limited to colorimetry methods. So we normally, depending on the naked eye or assisted camera, to monitor the color intensity of the reaction spot. But the problem is it provides only a yes or no or semi-consultative analysis. So we need more sensitive or more consultative analysis. So now we implement other type of transducers like electrochemical or fluorescent methods. But the problem is those kind of transducers require additional power source to eliminate a UV LED or the power electrochemical detection as well as other on-chip functions. Then what is the best candidate? What is the best power source for those applications? What we need is microwatt level power source. And I believe the battery is not that good for those applications because the battery is expensive and sometimes even wasteful for this single use and the low power devices. So I believe paper-based power source will be excellent candidate for those applications. So far, many types of paper-based power sources have been proposed. Paper-based electrochemical batteries, paper-based lithium-ion batteries, nano generator, and paper-based bio batteries. But the thing is, these paper-based electrochemical batteries and lithium-ion batteries have a toxic material, so it is hard to dispose properly. And also, lithium-ion batteries is expensive, and the paper-based nano generator requires complicated fabrication, increasing cost. So I believe the paper-based bio batteries will be the best because it is environmentally friendly and it is disposable, low cost, and also other type of body fluids like glucose or urine can be used as a fuel for harvesting electricity. And also this type of fluids have many anolytes to be detected. So in this research, my idea is to make a paper-based microbial fuel cell. So by using bacterial metabolism, we can harvest the energy. So in this case, we can use paper as a reservoir here. So let me briefly talk about some operating principles of microbial fuel cell. So when bacteria break down the organic food, the NADH and the NAD plus functions are repeatedly oxidized and reduced to generate the energy in the form of ATP. And for this reaction, they need electron acceptors. And normally oxygen is being used for this reaction. But if we provide a solid electrode like this, then we can capture this electron. So this microbial fuel cell, or MFC, is very similar to conventional chemical fuel cells, except that it does not need the catalyst in the anode. It uses bacteria. They can generate electrons, and also protons are generated. And those protons must transfer through this ion exchange membrane and reduce at the cathode. So when one electron goes, then one proton must go to maintain the electron neutrality. So we, for the first time, developed a paper-based microfilter, and I reported this at IEEE in the MEMS 2013 conference. So at the time, we used paper as an anode chamber, and we used the flexible carbon cloth as an electrode. And also we used the paper as a proton exchange membrane. At the time, we did some chemical treatment here. So when we dropped a bacteria with the media, it instantly generates a current. Yes. So the great thing about using paper in this case is that the paper has the ability to wick its solution, so it promotes the bacterial attachment instantly. That's why we got the power as soon as we drop the solution. But the problem is, as you can see, the power density and the current density was low, which is not enough for practical applications. So here we added more anodes in a single chip here. So we have 4 anodes. So each anode has the electrode and the paper reservoir, and we have 4 inlets, and each inlet for each anode. So for example, when we drop the bacteria here, and this hole is for anode 4, so bacteria go down all the way to anode 4, and this is a hole for the anode 1. So we need 4 droplets to derive this microbial fuel cell. So for proof of concept, we used the Schwann elasticity, and I used ferritinide as a catholyte, and we measured the potential difference between the anode and cathode through the external load. So this is test result. When we use a single anode in microbial fuel cell, the current density was about 82. But when we use the multi-anode MFC, it goes up to 211, and at the time the power density was about 28.4 microwatts, and it lasted about more than 80 minutes. So I believe that's enough for powering other paper-based biosensors, because those kind of applications need only 5 or 10 minutes for output. But there are some challenges. Well, actually, my idea was to use wastewater, actually, because wastewater has a lot of bacteria, and they have a lot of bacterial food there. So this is our next future research. So, and also I want to know if other type of body fluids can be used as a fuel. And also in this research, I use potassium ferritinide as a catholyte, which is toxic and expensive. So my next research is to use an air cathode, so we don't need to use catholyte. The ferritinide chemicals. And also here we use commercially available proton exchange membrane, which is extremely expensive. So we are going to use paper-based proton exchange membrane. So this is our recent result. So when I connected the 4 multi-anode MFCs in series, we could light up LED for more than 30 minutes. So that should be enough for other type of paper-based biosensors. So let me conclude my talk. So we developed a multi-anode paper-based microbial fuel cell generating 28.4 microwatts per centimeter square. And this paper-based microbial fuel cell is expected to be simple and easy to use power source for potentially single use in the diagnostic biosensor. So thank you for your attention. Any questions?